I think usually should be good. Okay, Julia's
Okay, you didn't let people in yet, right? Okay. I just wanted to spiel a little bit. Last we did with anatomy, we kind of ran out of time, low key. No, like we went over, like we had too much time left. Disregard everything I just said. But yeah, we had a lot of time left, so if that happens, I'm just going to spiel. Does everyone know their parts and everything? Probably by now, right? Thumbs up. Yeah, I won't spiel right now because we have to let people in a minute. But if so, if it comes to, just like try to explain as much as you can. Other than that, don't worry if like don't go too slow because I could cover like just speaking a lot. So yeah, you guys know I speak a lot, so at this point. But yeah, okay. It's nice to see all of your faces because it's usually just me on camera. But okay, in a minute we could start letting people in. Or how many people are in the room? Just wondering.
8. Wait, it's like 3. Should I let everyone?
Yeah, you could let them know.
Yeah. Hi guys, so we're going to start at in 5 minutes, so at 3:05. Like we're gonna wait until everyone else joins. Okay, so hi guys and welcome to molecular biology class. For today, we're going to go over an introduction to this class as well as molecular biology in general. So first, we're going to do some introductions. So first, the instructors will say their name, age, state, and their favorite ice cream. And then after that, all the students will go. So first, I'm Catherine, and I'm a sophomore, and I'm in California. And my favorite ice cream is probably mint chocolate chip.
Hello, I'm Maddie. I'm almost 16. I'm from California. I'm a junior, and my favorite ice cream is peppermint fudge ripple from Sweet Retreat.
Hi, I'm Yulia. I'm a sophomore in California, and my favorite ice cream flavor is vanilla. I know, kind of boring, but.
Hi, my name is Alexandra. I'm a senior. I'm from California, and my favorite ice cream flavor is also mint chocolate chip.
I'm Shrinjoy. I'm a senior. I'm in California, and my favorite ice cream flavor is matcha.
And although I am not an instructor, I'm Sophie. Nice to meet you all. I'm the CAO of STEMEY, and I just kind of oversee the class. I'm in New York actually, and my favorite ice cream is probably pistachio.
Okay, so can the students go in alphabetical order by first name? So like to avoid that awkward pause, I guess. So first, who's so whoever's name starts with oh my gosh, sorry. A, so that's no, Deepta. Deepa. Can you go?
That's my mom's name. I signed in from her account. My name is Ira. I'm 12. I live in California, and my favorite ice cream is strawberry.
Also, if someone's not comfortable speaking aloud, you can also type it in the chat if you'd like. So next would probably be Grace, if you want to type it, or anyone could type it in chat.
Sorry.
And then anybody can kind of go if you'd like. It's okay if you guys step on each other's toes and someone will just go, and then the next person can after.
Right, I guess I'll go. I'm Rishik. I'm 14 and I'm from California. And I guess my favorite ice cream flavor would be mango.
Hello, my name is Pollock. I'm 13. I live in Massachusetts, and my favorite ice cream flavor is cookies and cream.
Love that ice cream. I think a couple people didn't go, but if you're typing in chat, if you haven't done it in chat, then that's okay. But you could just put it in there if you haven't said it out loud.
Okay, I will go. So my name is Victoria. I am 16 and I'm from Ukraine. It's a country. And my favorite ice cream is, I think probably caramel ice cream. Yeah.
So hi, my name is Cornelia. I'm 17. I'm from Poland, and my favorite ice cream would be coffee ones.
Okay, thanks everyone. First I'll be going over like our class and first of all, the class will occur on Sundays from 3 to 4 at the same time as this class, but we will be sending out a form at the end of the class that asks which times you prefer other than this time. So yeah, look for that form at the end of class. Be optional and we won't have that much homework, but it will usually consist of worksheets, problem sets, or fun activities online that you can do on your own time. And we will answer all of your questions at the end of class or at the beginning of the next class. During class, we will incorporate a ton of fun activities as well, such as Kahoot, Scribble.io's, experiments, discussions, and more. And this class will be pretty unique from other classes because we'll be giving you a ton of opportunities and applications for the knowledge that we're going to instill upon you guys. So we will introduce many modern topics in molecular biology and some applications such as research projects or any other like modern topics such as COVID vaccines and such. And after you take this class, you will also be able to apply this knowledge to AP Bio or the SAT2 subject test or a ton of other biology competitions. We will be covering 4 units: biological molecules and cellular biology, which includes the membrane and organelles and other processes, metabolism, which includes photosynthesis and cellular respiration, and genetics, which includes cellular reproduction, genes, and heredity. And does anyone have questions about this class?
Everything is clear.
Okay, thanks.
Okay, so the main focus of molecular
Okay.
biology. So molecular biology is the study of the synthesis, structure, and function of various biomolecules and components of cells. So this can include things like water and macromolecules, so lipids, proteins, carbohydrates, and the nucleic acids, DNA and RNA. We also talk about cell membranes and its structure and how it functions and works in the cell. And we also focus on the organelles and their functions. Another thing that we focus on is cellular processes. So this includes things like photosynthesis, cellular respiration, metabolism, and the various chemical reactions that happen in cells, and also transport. So how materials are moved within cells and in and out of cells. We also talk about the cell cycle and division. Another main focus is on genes and gene expression and how gene expression is regulated by various factors and how the regulation of gene expression impacts cells and the traits that we express. So another main topic and focus of molecular biology is the central dogma, which is how DNA is turned into proteins. So as you can see from the pictures, this is how the central dogma works. So first, it goes through a series of steps. So DNA is first turned into mRNA through transcription, which occurs in the nucleus. And then mRNA is turned into the amino acid polypeptide chain through transcription. And the amino acid chains are then folded and modified to make the proteins. So the picture above is a picture of translation. As you can see, the mRNA is going through the ribosome to make the amino acid chain. So in this class, or in molecular biology, there's also a lot of research being done and a lot of technology that's related. So some things that we're going to be talking about in this class include technology and techniques such as DNA fingerprinting, stuff involving the environment, gel electrophoresis, cloning, and PCR or polymerase chain reaction. Molecular biology also is really involved in medicine, and This includes things like gene therapy and new drugs and treatments that are being created. Molecular biology is also really related to other types of sciences and other branches of biology, including biochemistry and genetics. And so in today's class, we're going to be talking about the history of molecular biology and some important people that were involved in molecular biology. We're also going to be talking about various techniques and technology that's used in molecular biology and the importance of these things and how molecular biology is applied in real life and in the medical field.
Okay, so I'll be talking about I'll be giving you a brief history of molecular biology. So first off, you probably got this by now, but it's extremely interdisciplinary, right? So it combines areas of genetics, biochemistry, and more. It also has a huge number of applications. So it originated in the 1930s through the realization that these fields investigate molecular structures within the cell, right? And the main difference, or I'll actually get into that later. So in the 1950s, we started exploring these molecular structures through techniques such as X-ray diffraction, which allowed us to gain more insight into how the molecules look. And from the structure that we could get from these diffraction techniques, we could also get some insight into their function and how they're actually important to the cell. For example, one of the biggest ones that you may have heard of is um, the structure of DNA, right? It's a double helix, and its structure is actually integral to its function, right? So if you're looking at, for example, the base pairs or things like that, that's where the information is stored. Or if you look at the backbone, that gives insight into how it's replicated. So it's things like that, right? So molecular biology also includes the importance of structure and how it relates to function, which is a theme that's actually present in all of biology, but especially so in molecular biology. Um, Next, we're in the late 20th century, where we could combine these fields of biochemistry and genetics in order to provide a biological explanation for how genetic information is actually passed on. So before that, biochemistry and genetics were considered relatively distinct due to their vastly different approaches. Right? So biochemistry looks at the interactions of molecules within the cell, so single molecule interactions, why they're important, things like that. Genetics looks at more big picture things. So they're looking at whole populations and how frequencies of traits are changing and things like that. But when they were combined, they provided a view into both the molecular and evolutionary bases of inheritance. And this combination is part of what we call molecular biology today.
Hi, so I'll be giving just a brief overview of some of the techniques used in molecular biology. And so I'll be giving the brief overview of molecular cloning, gel electrophoresis, polymersion chain reaction, and macromolecular blotting and probing. So molecular cloning is the creation of recombinant DNA molecules, and this has spurred progress throughout the life sciences. There are 4 steps of molecular cloning, which is shown by the image. So the first is isolation of target DNA fragments. Then the second step is Bing ligation of inserts into an appropriate cloning vector. Then the third step is creating recombinant molecules and the transformation of recombinant plasmids into bacteria or other suitable hosts. Another technique used is gel electrophoresis. This is used to separate DNA fragments. An electric current is applied to the gel so that one end has a positive charge and the other has a negative charge. This technique is commonly used in labs. The next technique is the polymersion chain reaction, or also known as the molecular photocopying. This is a method that is used to rapidly make millions to billions of copies of a specific DNA sample, which allows scientists to amplify it to a large enough amount to study in detail. This is done by denaturalization by heat, enabling primer to target sequence extension, and then out of the first polymersion chain reaction cycle. The next and fourth technique is macromolecular blotting and probing, and this is a method of transferring protons, nucleic acids, and ribonucleic acids onto a carrier, usually after gel electrophoresis. This nucleic and ribonucleic acids can be probed for specific sequences by complementary strands, while protein mixtures can be probed for specific proteins. There are many more techniques used that are very interesting in molecular biology, but these are just a description of some.
And now I'll be talking about why molecular biology is important. So first, it's important for molecular medicine, and molecular medicine is a broad field where physical, chemical, biological, bioinformatics, and medical techniques are used to describe molecular structures and mechanisms, identify fundamental molecular and genetic errors of disease, and develop molecular interventions to correct them. Molecular biology is useful because obviously you need molecular biology uh, to solve these problems and to cure some really important diseases. Another type of treatment is gene therapy, and this also uses molecular biology topics. Gene therapy is an experimental technique that uses genes to treat or prevent disease, and it focuses on the utilization of the therapeutic delivery of nucleic acids into a patient's cells as a drug to treat disease. Finally, molecular biology can be applied to plenty of real life situations. For example, in a study, the Hutchinson-Gilford progeria syndrome was uh, was a syndrome that involves premature aging, and this leads to death at the age between 10 and 14 years. And this study used topics such as the organization of the nuclear envelope, splicing, and gene expression, which are all in molecular biology. And after the study, it led to the discovery of the basic mechanisms behind this rare syndrome. And they figured out that the genetic basis in most cases is a mutation at the nucleotide position 1824.
Okay, so now I'll be going over some applications and the future of molecular biology. And I'm just going to go through a few of the many applications that exist with molecular biology. And this is just in hopes of like inspiring you to look into molecular biology because molecular biology, it can range from spectrum to spectrum. There's really a lot you can do with molecular biology. So one thing is DNA fingerprinting. And you know how like in the movies and crime scenes, people would like take pictures of fingerprints and compare it to the suspect's fingerprints. DNA fingerprinting is basically like taking it to the next level and it's more reliable. So basically like a swab of DNA is taken from the crime scene and it's compared to the suspect's DNA. And like Maddie mentioned earlier, this is with gel electrophoresis. So in the picture, it's this like gel box and these are DNA fragments and they separate by size. So number 1 on the very left, that's the that's the suspect's DNA, and then 2 through I think it's 9, those are all the different swabs of DNA. So you don't have to answer this, you can just think about it, but looking at number 1, which of the DNA fragments from 2 to 9 match the pattern of number 1? So you can just think about it. And if you look through, it looks like number 9 matches number 1. So that means that number 9, that's the suspect that was, and their DNA matches the DNA that was swabbed at the crime scene. So I found that pretty cool. And then, as you know, climate change, it's a very real problem, and molecular biology can be used to solve and help climate change. So some projects that scientists are working on is, for example, they're making plants that purify salt water. So as you know, we have a lot of water on this earth, but not all of it is fresh and clean and like you can't drink it, right? So they're making plants that purify this water and make it drinkable, basically. And then something else is they're making nitrogen producing microbes. What microbes do is they insert the nitrogen right to the roots of the corn to make it grow. And then another thing is methane consuming microbes. So methane is a greenhouse gas and it's really important. It's a really big factor of climate change. And so these microbes they consume the methane, which will obviously help climate change. And then I don't know if you know what Beyond Meat is, but in that little picture, in the middle, there's like a burger, and Beyond Burger, it's basically it's mock meat. So it's meat. It tastes like meat and it looks like meat, but it's not actually real meat. And when you cook it, little blood droplets appear on the burger, and the creators they did this on purpose. And how they made because it's not real blood, so how they made it look like blood is with biotechnology techniques, and they use plasmid cloning to insert the DNA of soy, and the soy created this red pigment, and they inserted the gene of soy into yeast so that the yeast when the yeast is used, it produces the blood that comes from soy, if that makes sense. And this is all thanks to molecular biology.
So now we're going to do a Kahoot review activity. And wait, I'm going to stop sharing for a sec so I can prepare. Okay, so everyone can just enter the pin and then wait, do you guys hear the music? And
Yeah, we hear it.
Join. Like if you guys want to join, you guys can also join. It's like, yeah, I'm gonna Okay, I'm gonna start now. If anyone else wants to join, you can just join the pin. Nice, good job. We're gonna cover all of these. Our curriculum will not be super difficult, but it will cover most of the topics. Good job, Srinjoy. Okay, so even though the word insert is in the inserted DNA, it's actually called recombinant DNA, and we will be going over this in the later weeks, so don't worry if you got it wrong. Yes, so as Alexandra stated, DNA makes RNA and then RNA makes protein, and that's the central dogma of molecular biology. Good job, guys. Yeah, it was originated in the 1930s. Nice. Srinjoy is still in the lead, and Palak is second. Pretty straightforward. Nice job, Rashik. As you may have noticed in the picture, it's inserting recombinant DNA into, I believe, a rat. I forgot. Okay, we have one more question left. The last one is a fun question. Nice job, guys. Okay, good. Everyone. Good job. And Rashik. Good job to all the winners, and Yulia and the smiley face. Um, we go back to the.
So we wanted to just share a couple of the units that you guys will be going over. So we're gonna pull up the. Oh yeah, if there's any questions, yes, of course. Catherine, I'm gonna pull up. Can you pull up the document of the overview of everything? You don't have to share it though. Do you know what I'm talking about or no?
Oh yeah, the curriculum.
Yeah. Just like so we could just like say like, oh, like this is what is going to be in the class, just to give you guys an overview of what you guys will be learning. To put you on the spot, sorry. But yeah.
I kind of went over it, but like we're getting it. Let me see. Wait, did you send the form out yet?
Oh yeah, someone is here. I'll send it out. So this form is just going to be just how you guys thought the first class was. It gives you an overview. We'll go over it actually. I'm just gonna send it in first. Oh, do you have it? Okay, because I don't know where the link. You got it.
Yeah, let me put the link in here. Oh shoot, okay. Yeah, so just fill out the form by this week, or just fill it out right now, and then using the results will determine whether to change the time or not, or like how to improve.
Something I just wanted to mention. I just wanted to go over some things that we have going on simultaneously. We also have STEMtober going on, and if you haven't heard about that, it's just another convention that we're doing from universities. Like we had a couple bio ones. Like I interviewed the professor from the biomedical engineering department at Johns Hopkins. So that's very interesting and definitely relating to this topic. So if you guys want to check that out, we posted that, I'm pretty sure, on the YouTube. So that's up there. And there are other ones coming up, but they're not that bio related, but there has been ones in the past, and there's also more to come. So I'm going to send the link in here. Here, I'm going to get it. So that's something interesting that we're doing if you haven't been keeping up with us on that. Also, I just wanted to mention how people might be like, oh, like why should we attend this class over other classes that people provide? the thing is that our classes and the style of how it goes and how every like class is planned is based off of what works for you guys, and that's like one of the most important things, because there could be a class with a harder topic where we just constantly answer questions about, like, say like we're stuck on a slide and we just answer a lot of questions based off of that slide. Like that's completely okay. Then it like could go on to the next class that we have, and we could discuss more about the topic. Or say we have a full day where we talk about homework or like what you guys had, and then go over certain problems and the homework. Like that's, it's completely up to you guys, and that's also on the form for how you would want the class to be split up. We could also have an open conversation about it if someone has a suggestion or whatnot. But the important thing is that it's not just a structured class, but it's you guys also controlling the class and how you guys will learn in your learning environment, and we could adjust to you. Also, there is one-on-one tutoring that is starting up. So if you need help with biology, whether it's in school or you just want to learn more about biology, and people could like give you homework separately also as well. So that's in the Discord as well. Is everyone in the Discord, by the way? If not, we could find a link for you guys so you guys can join. Here, I'm gonna send the STEMtober stuff also. But I think everyone's in it if they found it through this at least. Okay, this is the STEMtober link. But if anybody has any questions about anything we went over today, oh, I know something we could talk about. We could talk about everybody's experience. We could go to like the instructors first, like your experience with bio in general or molecular in general, and like classes you've been taking for the and like for the participants. Like you don't have to say that out loud, but for example, for me, I took AP Bio. I'm a senior, so I took AP Bio my sophomore year. And then I was going to attend 2 classes this past summer, but you know, COVID-19. So that didn't really work out very well. At one at Stevens Institute of Technology for biology, and then another one at Boston Leadership Institute in Boston for biomedical engineering. And I didn't get to attend those, which was very sad. But other than that, I just like do my own research. I'm part of another organization that we write like STEM articles, so that was fun also. And yeah, so that's like my experience. Does someone want to go next? And we could just like go to the participants also, so we could get an idea of just like where everyone is and like how experienced you are with like STEM and biology in particular.
Yes, so I'm a sophomore, and although I didn't really take it at school, I still studied biology, AP biology and AP chemistry, and I'm also taking AP biology, general like biology related courses in JHU and like online courses. And I also have conducted research related to biology, and I've competed in a few competitions as well. And yeah.
Does anybody else want to go of the instructors?
Okay, I guess I'll go next. So freshman year, I took biology, where we actually did use the electrophoresis gel. So that was pretty fun. And we actually got to do labs, unlike this year. And now I'm taking AP Chem, which does have a little bit of biology. But other than that, I just do research outside, and I'm part of several clubs at Lee that do mention molecular biology.
I can go next. So yeah, I took I took bio as a freshman, and we also did a lot of labs. And I'm currently taking chemistry, which has a little bit of biology in it. And I took online courses for biology at edX, and I'm also in various clubs, science clubs at school.
Did everybody go with the instructors?
Yeah, okay, I guess I'll go.
Okay.
So yeah, so I self-studied AP Bio, AP Chem. I also conduct research outside of school. I'm part of like a bunch of school clubs for biology. Currently, I'm actually taking like the course for AP Bio. Unfortunately, we don't get to do labs or anything, which is very sad due to this global pandemic. Um, I've also competed in various biology competitions, and I've taken like courses through like edX and Coursera.
So I have taken AP Biology, and I have also taken online biology courses through like edX and also at my local community college. And I've done my own research and stuff like that.
If anybody wants to say from the class, then you could go ahead if you'd like. This is just like an open discussion type of moment. Here, wait, I'm gonna go onto the form really quick and see if we have to clarify anything. Let's see. Yeah, so it also asks there like experience. If you don't have any experience, like that's completely okay also. That's what a class is for, so understandable completely. It also asks like what you're most likely, like what you're looking to gain from this, especially because it's like I said, like our environment is very like flexible rather. It's very flexible, so we could move around like what we want to do in classes. It's not really set to a strict schedule, and that's what makes us unique. So it has a bunch of like listings there, like of what you want to gain from this, which is great. It also asks if you want to join in the future, and would you prefer a 30-minute or an hour session, which is really good because you guys have a choice in that sense also. We just like having everybody involved because that just makes it easier and everyone could have a voice. And also like this is one of the big things I was talking about. So the formats of the class, like, oh, like you could have half lecture, half practice problems. It's just homework help, etc. And you could like put your suggestions in that. So every single class is obviously different. And we do have other classes if you haven't seen that. We have anatomy, physics, and then math ones next week. And yeah, and then it asks like what times you're available and asks for because we're always looking for how to get better and how to grow. So yeah, that's what I have to say. Does anyone else have anything to say? Questions, concerns, comments? Especially from the crowd. If you have any questions, this is the time to ask. And you could always PM or DM any of us on Discord. Most of us are probably usually on. But yeah. All right. Oh, like how someone mentioned before how they did gel electrophoresis. I also did that my sophomore year at what's called Cold Spring Harbor in New York, and that was really fun. Even though our gel got messed up, and they put like the images up on like the smartboard is very cool. And we also did DNA recombination also, which was cool, really cool. You guys will definitely learn about all of those things, of course, and don't be shy if you haven't learned about it yet, because this is what it's for, like I said before. Other than that, does anyone else have anything to say? I thought I'd just share my little story about what I've experienced also in AP Bio. Nobody? All right, I think we're good then. Thank you everyone for attending. Does anyone want to say anything from the teachers or instructors? No, we're good. Okay. So thank you guys for coming. We really appreciate it. Instructors, you could stay back for a little bit, and whoever's recording could stop it now.